So it is my pleasure to introduce Dr. John Majoris today as our seminar speaker. John from University of Texas Marine Science Institute, where he's a research scientist. Prior to that, he did a postdoc fellowship at Coast in Saudi Arabia. Prior to that, he did a postdoc at Boston University, where he also did his PhD with Peter Buston. And prior to that was at Florida Tech and has a degree in marine biology and aquaculture. John does field research in all over the world, from the Red Sea to Australia to Belize. And he's going to talk today about coral reef fishes, particularly Belize and larval behavior, but he also dabbles in sea anemone ecology occasionally, and we've co-authored a paper together and hopefully some more in the future. So let's all welcome John to Dizzle.
Yeah, thank you for that introduction. I'll just say my name is John Majoris. I am a research scientist at UT Austin's Marine Science Institute, and today I'll be talking a bit about some of my work evaluating the role of behavior and determining the pattern of dispersal in coral reef fishes. And in particular, this fish here, which is a neon goby, it's a sponge-tailed neon goby, so this big yellow structure that you see in the background here is a tube sponge, and they live inside of these tube sponges, and it's this really interesting connection. Okay, and so, Ben mentioned that I'm a marine ecologist. I'm kind of this when a variety of coral reefs from Saudi Arabia to Australia and Belize. And the other thing is that I'm also an aquaculturist, which means that I specialize in breeding and raising coral reef fishes. Okay, so in my research, I have found ways to combine these skill sets to address kind of three main research topics. And so that starts with developing and optimizing aquaculture protocols for coral reef fishes. I work with establishing broodstock, raising larvae, and optimizing how many larvae we can get through to settlement. I then use those lab-raised larvae to study the early life history traits and dispersal traits of coral reef fishes. And that's things like their morphology, behavior, and most recently, we're moving into transcriptomics. And then finally, I also conduct field research to kind of deepen our understanding of the role that larval behavior plays in driving population dynamics on coral reefs. So in my seminar today, I'm going to kind of touch a little bit on each of these three areas of my research. And I'll get started by giving you guys a bit of background about dispersal in marine systems. Okay, so the dispersal of offspring away from their natal habitat is this widespread phenomenon in nature. And it plays a really important role in maintaining demographic and genetic connectivity among distant populations. Okay, and this is especially true on coral reefs, where habitats tend to be highly fragmented, and the adults of many organisms can spend their entire lives within a few square meters of patch reefs. So this image over here is an aerial view of a reef in Australia. And these little outlines that you can see here are patch reefs. So that's the coral outlining a patch reef. And then in between these patch reefs, you have these expanses of sand. And so it's really amazing that, some adult organisms spend their entire lives within just a few square meters of this reef. Okay, and so in this case, the dispersal of offspring away from their parents is a primary driver of connectivity among populations. And this has really important consequences for the ecology, evolution, and conservation of marine species. And so for this reason, my research primarily focuses on the processes that influence patterns of dispersal in marine systems. And I primarily focus on studying coral reef fishes. And so I'll give you a little bit of background on their early life history. So most coral reef fish produce dispersive larvae that develop in the upper water column for about 35 days on average. And then they have to return and settle on benthic habitats, whether that's a coral or a different type of substrate. And so during this time, the larvae are thought to disperse widely, connecting distant populations and colonizing vacant habitats. And I want to emphasize that larvae are often incredibly small. So Here we have a developmental series of a green chromis, which you can see just a picture of the adults to give you some context here. And so these larvae are just 2 millimeters long at hatch. And you can see, there's not much going on here. You can't see any eyes developing. They're pretty early in development. And so as they go throughout development, they reach about 10 millimeters long at settlement. So they're still pretty small when they reach this phase of returning back to the reef. And so due to that small size, larvae were once thought to disperse passively by just kind of drifting around with ocean currents to reefs kind of far away from their natal origin. And, effectively imagine that you've set something about the size of an ant adrift in the ocean for 30 days. It seems pretty reasonable to assume that it would drift over a really long distance, right? But we now know that late stage coral reef fishes have a suite of developing sensory systems. Many have strong swimming abilities, and some can even orient their movement with respect to one or more different types of kind of sensory cues in their environment. So at this late stage of development, they can be pretty behaviorally capable. Okay, and so those traits, when you take them together, they suggest that these larvae probably aren't just drifting around in the open ocean, but instead they can actively influence their dispersal trajectory. And these active dispersal behaviors, they may not be limited to these late stage larvae. So studies using biophysical models to predict that early development of swimming and orientation behaviors can actually increase survival to settlement and help to retain larvae near their natal reef. So these kind of theoretical predictions from models suggest that selection will favor the early development of orientation behavior. But there's a problem. We know relatively little about when the sensory systems, swimming abilities, and orientation behaviors develop during these really early life stages. And that's because it's very hard to work with live larvae early in development, so throughout that early developmental stage. And few studies have actually quantified the dispersal distances of marine fish or marine species in their natural environment. Okay, so my research has primarily focused on addressing this gap in our knowledge. And kind of one of the critical questions in my work has been, when did larvae develop the sensory systems, swimming abilities, and orientation behaviors that could allow them to influence their dispersal? And my work is primarily focused on addressing these questions using this study system. It's called the line snout neon goby. It's a sponge-dwelling goby that's native to Belize, and it lives in these yellow tube sponges. So the majority of my research has taken place on the Mesoamerican Barrier Reef off the coast of central Belize. So if we're up here, this is Mexico. Belize is a small Caribbean country down here, and it's a really great place to study marine ecology because it has the second largest barrier reef in the world. So that's second only to Australia's Great Barrier Reef. And so our main study site was located off the coast here, right along the barrier reef. So we were working on this island called Caribou Key, and we had our research station right there. Okay, and so on this reef, the reef is primarily dominated by soft corals and sponges. And Elacatinus lori live and breed in this yellow tube sponge, which is called Aplycina fistularis. And when you look down into that sponge, you can see there's a male and a female with a clutch of eggs at the base of the sponge tube down here. So these little dots are a little hard to see, but they're laid on the edge of the inner sponge tube. So that relationship between the goby and the sponge makes it really easy to work with this species on the reef. Because really all we have to do is go out there and find these bright highlighter yellow flagpoles, and we're probably going to find our fish. Okay, and so as part of my work, I described the life cycle of this species. And so once the adults lay eggs at the base of the sponge tube, those embryos take approximately 7 days to develop. The larvae then hatch and they swim up into the water column where they develop for about 28 to 58 days before they then have to return to the reef and settle on sponge habitats. So these settlers, this is called a settler, then develops for a while on the outer sponge before it recruits into the sponge tube and eventually grows to reach adulthood. Okay, and so our research group was initially interested in measuring how far Elori larvae disperse away from their parents between hatching and settlement. Okay, and so we went along the reef and used this giant syringe here that you can see, and this thing is aptly called a slurp gun. And we can go up to the sponge tubes and suck the gobies out of their sponge tube to collect them and take a tiny tissue sample. And I'm going to show you actually what that looks like briefly here. Okay, so you'll see that kind of the tip of the slurp gun is inserted down into the sponge tube. They suck out the goby, pump it into a hand net. The other diver then takes the goby gently, turns it around so that its tail faces out of the net. They pick up their scissors and tweezers. They take a tiny fin clip, store that sample in an Eppendorf tube, all underwater, and then return the goby back to its tube. So I kind of think of this as a bit of an alien abduction. So these gobies, you know, this took all of 30 seconds, but, you know, we take that little tissue sample, it grows back in about a week. They survive this process. We've gone back and seen the same gobies over and over again. So we imagine that those gobies have a really good story to tell the rest of their friends. Okay, and so we went along the barrier reef and we sampled adults, so 3,000 adults at 3 focal populations. So you can see these boxes here were our adult populations. And then we also collected 4,000 settlers, or those late stage larvae that had just returned to the reef. And we did this along 40 kilometers of the Belizean barrier reef. So you can see all these points that we sampled. And so my collaborator, Cassidy De Aloya, used genetic parentage analyses to kind of connect offspring with their parents. So if you sampled a parent, say at this population here in the center, and then you found one of its offspring way up here, you then know the linear distance that that larva had dispersed. And when you have enough of these connections, you can graph the dispersal curve for the species. Okay, and so what we found is that peak dispersal occurs within the first few kilometers and then declines exponentially with increasing distance away from their parents. And this was with most of the larvae, you know, they only dispersed about 1.7 kilometers. And the maximum dispersal distance that we found was 16.4 kilometers, despite the fact that we had sampled extensively out to about 30 kilometers away from the parents. So we're pretty confident that this represents the dispersal curve for the species. Okay, so one of the predictions was that this pattern of dispersal can be generated by, you know, how long the larvae are drifting around in the water column before they return to settle on the reef, right? And so this is called their pelagic larval duration, or PLD. But we're actually able to look at that. So there's a tiny bone in the ear of fish. It's called an otolith. And the way that calcium is deposited in this bone is in rings. And so early in development, these calcium rings are deposited on a daily basis. And so you can actually take that otolith out, polish it, and look at the rings to tell how many days it took to develop in the water column before it returned to the reef. So we can tell its age at settlement, kind of like tree rings. And so we did this with our samples. And what you can see here is that, you know, larvae that were dispersing or developing for shorter periods of time were not dispersing shorter distances. And larvae that were developing for longer periods of time were not, you know, dispersing really long distances. So there wasn't a real clear relationship. This is a kind of flat relationship there. And so this suggests that the pattern of dispersal is unlikely to be, you know, explained by how long these larvae are developing in that upper water column. And instead that there may be, you know, a role for larval behavior and dispersal traits to, you know, better understand or predict this pattern. Okay, and so I was interested in determining when Elori larvae develop the sensory systems, swimming abilities, and behaviors, you know, that could help to explain this kind of restricted pattern of dispersal for the species. And so to address these questions, and especially those about larval behavior, we need to establish a source of live larvae. And so this has traditionally been a really challenging thing to do. Okay, and so previous studies of behavior have kind of relied on you collecting late stage larvae in this device here that's called a light trap. And so this is a kind of mesh cylinder here, and it's got these conical shaped entrances. And when you suspend a light in the center of that chamber, the larvae are attracted to that light, kind of like moths to a flame. And so you can collect late stage larvae using this technique, but it's really selective. Only a few species are really strongly attracted to that light, and only really late in development. So we weren't able to really understand what's happening earlier in the larval phase. But aquaculture provides an approach to kind of work with larvae throughout the entire larval phase, from hatching through to settlement. And we've gone from being able to raise just a few species of coral reef fishes to over 300 different species in about the past 10 years. So this, these advances in aquaculture have really opened up a kind of unique approach to being able to study larval behavior and larval development throughout the entire larval phase. Okay, so to develop an aquaculture protocol for E. lauri, I built 2 rearing facilities, one in our lab in Boston, which you can see here, and one at our field site in Belize. And these facilities allow me to develop the techniques for raising this species in captivity. Okay, and so to do that, I established breeding pairs of E. lauri in the lab. And luckily for me, they take to these kind of PVC tubes, kind of like sponge surrogates. And after spawning in the tube, so this is a male with a clutch of eggs here. They're kind of these tannish color when they're early in development. Those embryos take about 7 days to hatch. And so on the night before they hatch, we move them to a separate rearing bin so they can hatch out in a kind of dedicated tank. And we feed them a variety of plankton, things like rotifers, copepods, and artemia. And kind of using that approach, the larvae will develop for about 30 to 35 days in the lab before they reach settlement. And so, really nailing down that aquaculture protocol gave me a unique opportunity to work with larvae throughout development, from hatching the whole way through to settlement. And you can see on the side here, I was able to work with both live specimens and preserved samples and take some really nice photos of their development. So I just want to walk you through a couple of the features that you see in these photos. So this is a 6-day post-fertilization embryo. So that's 1 day before it's about to hatch. And one of the things that really stand out to me are these big, well-developed eyes. And then if you look a little farther back, there's these 2 spots. Those are the otoliths, or the ear bones that I mentioned earlier in the presentation. They have a large yolk sac, which gives them an energy reserve that allows them to develop. And then they have this little inflated swim bladder. Okay, so once those larvae hatch, you can see, this yolk sac has almost been depleted. So they have about 12 to 24 hours before they fully use up that yolk sac and have to begin feeding. So they have to begin feeding pretty rapidly after they hatch. The other thing you can see is, or maybe vaguely see, is that they don't have traditional fins, right? So they have this kind of median fin fold or flap of skin that kind of runs along the outer perimeter of the body. So they're not as capable of swimming at this point. But as we go through development, you can see that these fins are, you're starting to see the paired dorsal and anal fin, something that looks more like a traditional caudal fin. And those things progress in development until the larva reaches the age where it's about to settle. And one of the cool things that you can do is that they have this fused pelvic suction cup. And so that fused suction cup is unique to gobies, and it actually allows them to stick to the substrate. So they actually can stick to that sponge when they settle. Okay, and so having specimens at all stages of development allow me to take a closer look at the sensory system. And so in collaboration with Jackie Webb's lab at the University of Rhode Island, I used a variety of morphological techniques, including vital fluorescence staining, histology, and scanning electron microscopy to study all of their sensory systems. throughout development. And I'm just going to focus on a few of these in more detail today, ones that may be related to their dispersal. Okay, and I'll begin by taking a closer look at the lateral line. So the lateral line system is a sensory system that allows fish to detect water flow around their body. And it's made up of a series of sensory receptors that are called neuromasts. And so I use vital fluorescence staining techniques to highlight individual neuromast receptors, which you can see up here in these photos glowing in orange. And then I also used scanning electron microscopy to give us a closer look at the structure of those neuromasts. And so kind of the main takeaways here are that throughout development, so at hatching, they have just a few of these neuromasts on the head. And you can see that as we progress through development, those neuromasts are proliferating, and then they begin to organize into well-defined lines on the head and actually also on the trunk of the body. And when we take a closer look at these neuromasts, what we can see is they have kind of this diamond shape. So these lines here are the kinocilia of the hair cells that make up the neuromast. And typically these hair cells are embedded within a structure that's kind of this gelatinous structure. It's called a cupula. And the cupula is removed during the preparation steps for SEM. But in this photo here on the end, you can see one cupula that made it through that process. There's something really interesting about it. It's kind of got this paddle shape with these kind of wing-like projections on the edge. And so this is sort of a unique structure for cupulae. Typically, in a lot of other fish species, these cupulae are kind of rounded. And so when the cupula is deflected by water flow, it stimulates those hair cells and they send a signal to the brain, right? So that's how the receptor is triggered and sends a signal to the brain. And so this kind of paddle shape, we think, provides a greater surface area so that there may be higher sensitivity to water flow around the body. Okay, so next we'll take a look at the ear or the auditory system. So the auditory system begins with two otolithic organs, the utriculus and the sacculus. So this is the utriculus and the sacculus. And then around day 10, there's a small additional otolith that develops called the legana. And so kind of one of the things to take away from this is that, these otoliths, they're increasing in number, but they're also increasing in size. So you can see gobies have this large square saccular otolith. So you can see that in these live images of the larvae, but also in these cross sections through the sacculus down here. So this kind of maroonish red dot here is the remains of the otolith, and it's sitting on top of a bed of sensory hair cells. So it's called a macula. And what you want to notice here is, again, as we go through development, that otolith is growing in size. And also the size of the sensory macula is increasing, and the number of sensory receptors in that macula is increasing. So what we think this means is that, as these otoliths increase in number and the size of the sensory system also increases, these larvae will become more sensitive and able to detect sounds in their environments, specifically ones from coral reefs that could allow them to find their way back to that reef habitat. Okay, and so finally, we'll look at the olfactory system, and this one undergoes some pretty dramatic changes as the larvae develop. So at hatching, the olfactory system is made up of these two kind of flat patches of olfactory neurons, and you can kind of see those here. And then with these cross sections through those olfactory neurons, you can see they have sort of a simple flat structure. So kind of a real smooth, there's no kind of convolutions or folds that would increase the surface area. And as they develop those sensory systems, they're getting larger. The neurons are kind of they're declining into a kind of pit-shaped structure, right, that keeps growing. And then finally, they become fully enclosed to form the incurrent and excurrent nares. And the reason that's interesting is once those nares form, the larvae or the settler can actually kind of move water over the sensory epithelium. So they can pump water through their nose. And before that, the larvae are relying on kind of passive ventilation as they swim through the water. So they are kind of forcing their way through the water, and that's what causes the chemical cues to interact with the neurons. So this is pretty interesting because, they can't really ventilate their olfactory system until very late in development. So one of the kind of predominant thoughts or hypotheses about how larvae find their way back to a coral reef is using olfactory cues. And so these larvae are not going to be very capable of using their olfactory system until right at the time of settlement or shortly after settlement. So olfactory cues probably aren't going to play as important a role in their ability to find their way back to the reef. Okay, so kind of to summarize this section, so all of the major sensory systems are present at hatch. And while the anatomy and pace of development is kind of similar to other fish species, Elori have a couple of kind of characteristics that are unique to gobies. And this includes the proliferation of neuromass on the head and trunk, this large square saccular otolith that I mentioned, and then kind of the simple, smooth structure and late stage enclosure of the olfactory system. So this morphological analysis suggests that the sensitivity of the lateral line to water flow around the body may be enhanced, and also that the ear may be more sensitive to reef sounds. But really, they're not going to be using olfactory cues as effectively as some other species. Okay, so now that I had a better understanding of when larvae develop each of their sensory systems, I conducted experiments to determine when larvae use their senses to kind of find their way around in the ocean. And in particular, these experiments looked at their swimming abilities, orientation behaviors, and settlement behaviors. Okay. But I always like to stop here for a moment and kind of consider the challenge that these larvae are facing. So as a diver, whenever you swim a couple 100 meters off the reef, you're kind of confronted by this wall of limitless blue ocean in all directions. And unless you have a compass mounted on your wrist, it's pretty much impossible to find your way back to the reef. It's really disorienting. And so, what I'm suggesting is that these tiny, ant-sized larvae somehow have the ability to find their way around in this kind of what appears to us to be a featureless blue ocean, right? So this is a pretty challenging task. Okay, and so to test the swimming and orientation behaviors of Elori, I use this device called a drifting in situ chamber, or DISC. And this was a device developed by Claire Paris at the University of Miami. And so what the DISC is primarily, it's an acrylic behavior arena. So this central compartment here is an acrylic arena, and below it, we have a package of sensors, including a camera and some things that monitor water quality and position. And so we can deploy this system under semi-natural conditions, meaning that it's offshore where larvae would naturally be developing, and we can observe the behavior of these larvae in the open ocean. Okay, and so here's an image from the camera mounted underneath the disc. And so if you kind of squint pretty hard, you can see there's a larva in the chamber right here. And so let me get this video going. So a diver goes down, they deploy an individual larva in the disc. They then get out of the water, and the larva's behavior is recorded for 20 minutes. After that 20 minutes, the diver returns, collects the larva, and replaces it with another, and the process repeats. And so using these videos, we can track the position of the larva in the disc. And from this, we can see this track is kind of all clustered on one side of the chamber. And we can determine the larva's swimming speed, turning angle, and mean bearing of orientation from these tracks. Okay, so I went back to the reef. I raised larvae in my field-based wet lab. I deployed them in the disc around 500 meters offshore from their natal reef. And throughout our 4-month field season, I deployed larvae every 2 days throughout development from 2 to 30 days post-hatch. And I had about 120 deployments. And so you can see we had a pretty nice spread of data across all ages of development. And so let's take a look at this data. So here I have the mean swimming speed and mean turning angle by age and days post-hatch. And what we can see here is that, these larvae, ultimately they're swimming pretty slow. So that's 1 centimeter per second at their max, right? But a takeaway here is that these larvae can swim very early in development and that their swimming is improving throughout development. We also see that their turning angles, so how extreme of turns they're making, decreases with development. So when you take those two things together, it suggests that they're swimming both faster and straighter or more linearly as they develop. Okay. And so here I'm pulling in data on the maximum swimming speed of larvae that were tested in a swimming flume. And swimming flumes are basically treadmills for reef fish larvae. So here I'm showing you a clownfish because they're a little bit easier to see than goby larvae, and they're a little cuter, actually. So let me click on this for you. Okay, so this is an 8 millimeter larva, and it's swimming at about 12 centimeters per second. And I can test the larva's maximum swimming speed by kind of gradually ramping up the speed in the flume until the larva effectively falls off the treadmill. So it kind of crashes on the back mesh here. Okay, and so using this flume, I swam Elori larvae at 0, 10, 20, and 30 days post-hatch. So I could test their maximum swimming speeds. And so what I found is here I'm comparing swimming speeds measured in the disc to those measured in a swimming flume. And we can see kind of a similar pattern throughout development. The swimming abilities are increasing throughout development. But we'll notice that in the swimming flume, the larvae can swim at about 10 times faster speeds than they're kind of naturally swimming in the disc. So they have much greater swimming abilities than we're seeing when they're out in the disc. And about halfway through development, they're reaching speeds that exceed the mean current speeds that we measured in the study area. So they're able to swim faster than the mean currents. Okay, and so to put that into context compared to other species, here I'm pulling in a figure from my collaborator, Jeff Leese, and it's plotting the swimming speed by size for different families of marine fish species. And so when I overlay Elori larvae swimming speeds, we can see that for their size, they're pretty slow. They're way down here among the cold water species, when most other coral reef fish are up here in red. So this was pretty surprising. When I had this result, I was thinking, okay, these larvae, they're staying really close to home. To avoid being dispersed by currents, they're probably these super athletes that are swimming really hard and trying to get home. They're able to fight against the currents. And ultimately, I found out they're pretty dopey. So this really threw me for a loop. And it made me go back to my data and think, okay, Elori are terrible swimmers. So what other behaviors might they have that help them stay close to home? And so I began to analyze that disc data to look at their orientation behavior and think about how that could help them stay close to home. And so this is just a reminder that from those tracks that we can measure in the disc, we can determine the direction that the mean direction that the larva is heading. Okay. So here we have plots representing the data for 4 individuals. So I've plucked kind of individuals randomly from our data set at 2 days, 10, 20, and 30 days post-hatch. So these bars represent the frequency distribution of the larva's position within that chamber, and the line represents the mean bearing that larva was heading in. And so what we can see is that each of these larvae at each of these stages of development was able to hold a significant bearing. So they were able to swim directionally. And in fact, I found that 78% of larvae oriented significantly, and the proportion of that oriented didn't change with age. Okay, so what that means, and this is really amazing to me. So these larvae, right about when they hatch or a few days after they hatch, have the sensory systems and swimming abilities that can allow them to find their way around in the ocean, that can allow them to actively orient and determine which way they want to go. And so that begs the question, where are larvae going? Or to put it another way, what are they swimming towards, right? And so there are several alternative hypotheses for the goal of larval orientation. And I've put together this kind of conceptual framework to help walk you through some of those ideas. And so this line here is meant to represent our transect along the Belizean Barrier Reef. And these numbers represent some hypothetical disk deployments. So the location of a few different disk deployments. And the arrows represent the direction that I predict that the larvae would swim under these different conditions. And so right away, we can kind of roll out the idea of passive dispersal, right? We know that these larvae have the ability to swim and that they're able to orient their movements. So they're not dispersing passively. If you take anything away from my seminar today, it should be that larvae are not dispersing passively. They have these different behaviors that allow them to influence their dispersal. Okay, and that leaves us with a lot of other hypotheses that can be really interesting, right? Okay, so larvae could employ a single strategy. For example, they may all be swimming towards the west in Belize, and that would carry them back towards the barrier reef. That would be a pretty easy strategy. They can employ a condition-dependent strategy. So for instance, they could swim offshore early in development and then back towards shore later in development. They could employ a context-dependent strategy. For instance, they could be swimming towards a similar location or environmental cue. For instance, they could be trying to home towards their natal reef. And finally, they could, all be employing different strategies. And in that case, the data would be pretty messy. So let's take a look at what I've got. So in this figure, each point represents the mean bearing of an individual larva in our study. Unfortunately, because you can see those are all distributed pretty randomly around this circle, these larvae don't agree on where they should be going, right? And when we subset this data and bin it by age groups from 2 to 10, 12 to 20, and 22 to 30, we can see, again, these points are all randomly distributed around the circle. So these larvae aren't sharing a common strategy, right? And we looked at all of these different hypotheses and found, they're not employing they're not following a constant bearing, a conditional bearing, swimming towards the same location or towards any environmental cue that we could test. So kind of frustratingly, this question of where are larvae going still remains, and it's going to take a bit of additional work to figure out. So let's return to these hypotheses and think about, what may be going on here. We can rule out passive dispersal, right? And in this context, these larvae don't seem to be using the same strategy. So it suggests that there's a lot of intraspecific variation in larval dispersal strategies in this species. And so it's going to be fun to kind of pick apart that hypothesis. Okay, so now we know that larvae are using their senses to find their way around in the ocean. And as they reach settlement, these larvae have to metamorphose and settle out of the water column onto benthic habitats. And so I was kind of interested in understanding, how larvae choose sponge habitats at settlement and how settlement behaviors influence the distribution of the species on coral reefs. Okay. So to observe the distribution of settlers on sponge habitat, I went back to the transect on the barrier reef and I recorded the presence or absence of Elori settlers on 2 of the most abundant tube sponge species. So this is the yellow tube sponge here, and this is called the brown tube sponge, Agulus conifera. And we chose that sponge as a comparison point because it has a similar morphology and comes in a variety of similar sizes, so similar tube sizes. So here we have the probability of settlers occurring on a tube sponge by the maximum tube sponge length. And what we can see here is that settlers were more likely to occur on large yellow tube sponges than they were on small sponges or brown sponges. Okay, so that kind of gave us an idea of their distribution. And so we were interested in understanding or determining whether the distribution of settlers on sponges was determined by variation in their survival on these different sponge species. So maybe they survive better on yellow sponges, or by habitat preferences at settlement. So maybe they're choosing yellow sponges when they settle. Okay, so To determine whether cellars survive better on different sponge species, I went back to this transect and removed the cellars from 60 yellow tube sponges and 60 brown tube sponges. And then I collected cellars from outside the transect and seeded them onto those vacant sponges. And then I went back and used scuba to survey these sponges every 2 days or 1 to 2 days and recorded the presence or absence of that cellar on the sponge. Okay, and what I found was that cellar persistence was also predicted by the sponge species and sponge size. So here we see that over time, the proportion of cellars that remained on a tube sponge was highest on these large yellow tube sponges, rather than these small yellow tube sponges or brown tube sponges. So that fits really well with the observed distribution, right? But consistent variation in survival among habitat types should favor the evolution of habitat preferences at settlement, right? So if larvae are able to choose the right or the best sponge species that would improve their survival, then that should be selected for in the population. Okay, and so to determine whether settlers prefer certain sponges at settlement, I collected settlers from sponges on the reef and then tested their habitat preferences in a circular arena in a kind of sandy area of the back reef back here. Okay, and so the arena was made up of 2 alternating habitat types for a total of 6 habitat choices. And after a 2-minute acclimation in the middle of the arena, the settler was released from the center and allowed to choose freely among these habitat types. And then I recorded the first habitat type that they made contact with, so their first choice. Okay, and so using the arena, I was able to test a variety of different kind of comparisons, right? So I was able to test yellow versus brown sponges, large versus small yellow sponges, multi versus single tube, and adult occupied versus unoccupied. So sponges that I had kind of seeded an adult fish into. Okay. And I also took this one step further for each of these comparisons and looked at whether settlers were using visual or chemical cues to make their decision. So by placing a clear cover, this is a clear plastic tube, over the sponge, I was able to provide visual cues while blocking chemical cues. And so this pink dye I injected in here kind of shows you that the tube was retaining the dye, so those chemical cues from the sponge weren't being emitted. In contrast, these mesh covers they standardized the visual cues, but they let those chemical cues come through. So you can see that the dye is kind of moving through the sponge. Okay, and so using the choice arena, I found that settlers prefer yellow over brown sponges in both the all cue and the visual cue trials, but not during these chemical cue trials. So whenever I had kind of standardized the cues using that mesh structure. And when comparing the large versus small yellow sponges, I found a similar result. So they prefer the large sponges using all cues and visual cues, but again, not during chemical cue trials. So there's this kind of pattern emerging, right? Okay, and so finally, I found that settlers showed no preference for multi-tube sponges over single-tube sponges or those that were occupied by adults. Okay. So just to kind of summarize these results, I found that settlers prefer large yellow sponges over small sponges or brown sponges, and they appear to be making these decisions using visual cues, but not using chemical cues. And that's sort of all aligns really well with their kind of sensory systems that we talked about earlier in the conversation, right? So those olfactory system doesn't really come online until late in development. And so when you take these things together, it suggests that differential persistence among these sponge habitats, so whether they can survive better on some sponges versus others, has favored the evolution of habitat preferences at settlement, that ultimately these pre and post-settlement processes kind of shape the distribution of the goby population on sponge habitat. So it's kind of a really cool kind of full circle analysis there. Okay, and so I set out to address this question of, when do larvae develop the sensory systems, swimming abilities, and dispersal behaviors that could allow them to influence their dispersal? And I think this kind of collection of projects gave us some really interesting initial results. And so we found that all of the sensory organs are present at hatch, that larvae swim both faster and straighter throughout development, and that most larvae, up to 78%, oriented, and settlers prefer specific sponge habitats at settlement. So they have these complex behaviors that they can employ. So when you take these together, the results suggest that E. lori larvae are equipped with all of the things they need, the sensory systems, the swimming abilities, and the behaviors that allow them to influence their dispersal. And it's not just late in development when they're settling, it's throughout their entire larval phase. Okay, and so these behaviors may help to explain that kind of short distance pattern of dispersal that we found for this species. Okay. And so I think this work has kind of broader implications for natal dispersal and connectivity in marine fishes. So ultimately, I think I made it clear that these gobies, they're not that special. Their sensory organs are pretty standard for different marine species, and they're effectively terrible swimmers, right? But despite all of that, we still found that they can orient their movements beginning really early in development. So it seems likely that a lot of other species, especially those with those really fast swimming abilities, may also be able to influence their dispersal throughout the majority of their larval phase. Okay, and so with advances in aquaculture techniques, the rate that we're able to raise new species, we now have the ability to raise a variety of coral reef fishes. And you can see, from a couple of the examples here, they vary wildly in form. So their morphology can be crazy and spiny. They can be really simple. They can have these long trailing fins that may either help or hurt their swimming abilities, right? And so there's a lot of intraspecific variation in dispersal traits. And so that's going to be interesting to dig into and to see how their sensory systems, swimming abilities, and behaviors, help us to better understand the processes that shape population connectivity in marine systems. And this is a really important concept because, the distance that we're spacing marine reserves is based on kind of random guess knowledge at the moment. So if we're able to understand the processes that determine how far these larvae disperse, we'll be able to better space our marine reserves to help conserve marine populations. And finally, I'd like to acknowledge a lot of people that participated in this work, especially the members of the Boston lab, Claire Paris's lab at the University of Miami, and Jackie Webb's lab at the University of Rhode Island. And finally, my funding sources, NSF, Boston University, and the American Museum of Natural History. Okay, and with that, I'm happy to take any questions. Yeah.
I was really great talk. So cool. I learned so many things. And I was wondering, why do the settlers stay on the outside of the sponge so long?
Yeah, we've been wondering that a little bit too. Some of it is because adults exclude settlers from the inner sponge tube. So that's one reason they kind of get pushed out onto the outer sponge. But even on vacant sponges, they're often on the outside of the sponge. So it's probably something, my best guess would be it's something related to, whatever they're feeding on is probably happening more often on the outside of the sponge than on the inside. The yellow sponges are also really textured. So they've got these ripples on the outside, and the settlers can kind of tuck into those crevices really well compared to the adults. So they're likely being, sheltered pretty well, even on the outside of the sponge, given their really small size. Yeah. Yes.
That was really interesting stuff. One question I have is, so if they've got about a 30-day larval phase in that system, how far will the passive particles disperse?
Yeah, it's the whole way up into Mexico. Given some of our collaborators have done a little bit of modeling on passive dispersal. Yeah, they could be
really not talking about.
We're talking
a kilometer instead of going 100 or more.
Yeah, around 100 kilometers. So 50 to 100 kilometers was sort of the range that they expected from those models.
Wow.
Yeah. Yes.
I was wondering with your collection device with the light sticks in there.
Yeah.
Not a nocturnal component of this behavior that you're describing here.
Yeah. So We are working on, yes, those are deployed at night, because that's when you get the most attraction to that light. And larvae have a lot of behaviors that are, that change throughout the day, throughout the light cycle. So they migrate vertically in the water column at night, so they come towards the surface typically. And they also seem to, settle at night. So most larvae are migrating onto the reef and choosing habitat at night. At least that's the predominant theory. But one of the challenges is to be out on scuba observing settlement habitat in the dark, right? So that these studies kind of assume that larvae arrive, out of nowhere the next morning. So to answer your question about, how do these things change? How can we assess that? We're beginning to develop a disk that has infrared lights on it so that we can test nighttime specific behaviors and see if those change the behaviors and the orientation pattern of the larvae.
I was a little surprised that they seem to just go in all directions, right? It's like they
Yeah.
take a fan and they go relatively see the big yellow sponge, right?
Yeah,
So, but they do have a long larval duration, right?
right,
So they could disperse farther that choose not to, right?
correct.
Do you think if you went to a system where the reef is healthy, that might be different? In other words, that they, like if you hatch on a reef and leaves, you can almost sit in any direction and you're gonna find a big yellow sponge.
Right.
That might not necessarily be true in other.
Yeah, that's a great question. And it's something that these kind of aquaculture techniques have allowed us to start pitching experiments for, right? So we're able to work with these larvae now. We know more about their behavior under natural context, which is really rare. This was the first study that looked at, the entire larval phase out in situ. So sort of next steps are to look at what things trigger alternative dispersal strategies. So, some individuals might be employing a strategy that keeps them close to home, so that helps them stay close to their natal reef. Whereas others might be employing strategies that just, allow them to disperse over long distances. And there's a variety of kind of theory and behavioral ecology and dispersal ecology about how evolution would favor those different strategies, and whether you would employ mixed strategies, whether parents can induce specific dispersal strategies through maternal effects or some other genetic components. So yeah, there's going to be a lot of interesting follow-up work, and that's sort of our next phase, is to look at that kind of variation in strategies. Yeah.
I'm sort of curious, what sort of logistical challenges come with culture these reef fish facility?
Yeah, lots. So to build a facility for culturing reef fish on a field station on an island in the middle of Belize meant that I had to pack every single PVC fitting into my airline bags, and I had 10 checked bags that year. To be able to build that wet lab from start to finish. So that was one of the major challenges, is even just getting the materials there to do the project. And that included live food cultures. So we had to have rotifer cultures imported from the US, and then be able to culture those in the field under conditions that were less than stable at certain times because of temperature. Then we got hit by a hurricane in the middle of the project, and I had to build a tarp that went the whole way around the lab, and then just hope that the larvae survived, because this is a 30-day investment, right? So every data point that we got late in development took us 30 days of loving care to raise these larvae to that point, right? So it was challenging. Yeah.
Springboarding off of that, you have larvae that presumably have a higher survival rate in captivity than the natural wild ones. And you're basing your behavior results on individual larvae's behavior.
Right.
Can you be confident that those weren't just extra dopey larvae that would not have survived in the wild, compared to wild behavior?
That's a great question. I've actually, mostly when I get that question, it's flipped and they say, okay, have you produced super larvae because you're feeding them all, all these prey items and they're in a safe environment and they, can grow under ideal conditions. So they can go both ways, right? In that question. And so right now I have to say, I'm not 100% sure that there's going to be a ton of mortality in the field, right? So only about 0.001% of larvae survive. And so that's a really huge bottleneck, a selection bottleneck that is going to, choose some individuals that are, ideally the most fit to survive, right? So what traits those individuals express is going to be hard to figure out. But we're starting a project now. I've pitched a project to look at the underlying genetic components or mechanisms that generate these variation in larval traits. So variation in morphology and behavior and physiology that may affect their dispersal distances. Then we have samples from the field that we know the actual dispersal distances of that we can resequence as well. And we're hoping that we can then make a connection between the traits that in their underlying genetic components and predict the traits that the individuals that we know their dispersal distances, we can actually predict their traits and see, if you stayed close to home, were you expressing a certain set versus if you dispersed over long distances, were you, expressing a different set. So we might get there, hopefully. Yeah.
So another way to look at the connectivity is with inflation genetic support.
Yes.
And so I was wondering because it's often it's hard for in the first place to have a good modeling of the ocean current in one sense. And then it's even more rare to have an idea of larval behavior and things like that. So
right.
I think you are you are getting super close, and maybe not super close, but you are making super good progress in that direction. Sorry. And so I was wondering if you have maybe if you feel plan to, and also what's super interesting, I think, is that you will have that on many different species at the same place.
Right.
So if you have any plans of comparing all of this and integrating some genetic diversity and differentiation data in a model that would combine larval behavior and current at that case to look at how much you can explain in terms of the pattern of genetic diversity.
Yeah, there's been a lot of population genetic work done by I mentioned my collaborator Cassidy De Loya, and it's been really interesting. So there is spatial genetic structure at about a 20-kilometer range. So some of the atolls offshore from Belize are spaced at about 20 kilometers, which is just outside of the range that we predict the larvae are dispersing. And there's pretty clear spatial genetic structure among those populations. So that's aligning really nicely to model that then and to integrate these things into kind of eco-evolutionary biophysical models. It's a lot of data to put in, and we're working on that actually at this point. Some other groups have had success in Australia integrating behavior into biophysical models and then predicting their dispersal, their measured dispersal curves that they measured using genetics. So it's clear that behavior as a component of models can be really important and help us make better predictions of the genetic component, right? So we're hoping to get to that point with this system as well. And that's kind of the next step here. So that question over here, yeah.
Yeah, awesome talk. Okay. Really interesting. It's triggered all kinds of things I want to talk about later.
Cool.
But I'm also interested, how have you started looking at, you mentioned these different sensory systems being developed really early.
Yes.
Auditory.
Yeah.
And there's olfaction. So how do you know that it is visual and not auditory? Because those things are linear, probably.
Right.
Have you had a bit of a look at that yet?
So we're in this project, no, we were trying to see, okay, just, when given everything they've got, when can they start to employ these behaviors? And I guess maybe I forgot to mention that, coming into this, our prediction was that, throughout a 30-day larval period, they're not going to they're not going to be able to do anything in the first 10 days, maybe, right? That's what we thought. And so it was really surprising. We thought, midway through development, they'd start to have these sensory systems that were more functional and swimming abilities that would allow them to maintain a directional bearing. But to find out that they're doing it from the start was a real surprise. To start picking apart the sensory systems and which sensory systems are allowing them to do that is going to be a little more challenging. We've done things like start to manipulate sun compass cues. So solar cues are a way that a lot of animals navigate. And so we can manipulate those by blocking them out or, changing their position, by clock shifting the larvae. And so we're starting to work on that. Auditory cues have been a little bit harder to eliminate because they're kind of ever present in a natural environment. So we're still working on how best to do that, but we can do playbacks, and it does seem like larvae are responding to playbacks from hydrophones. And then the kind of current cues are interesting because there is some signal in our data that larvae are orienting with the current. So they're, as the ebb and flood changes, that the larvae are kind of facing into it and just kind of going back and forth potentially. But we had a setup for the disc that shouldn't have allowed them to detect that movement. So we don't actually know how they were detecting that current change. So there's other kind of components to this that are going to be really interesting to pick apart to say, okay, what are the really important cues? Are they olfaction? Are they auditory? Are they visual? And, begin to say, for different species, even what those important components are. And it's probably going to vary a lot by species. Yeah.
Were you recovering like the larvae upstream and downstream from their like original location? So like, they were swimming against the current?
Yeah, yes. Well, so there was the disc drifts naturally with the current. So depending on the day, the current will determine where the disc is carried. And sometimes it gets carried north most of the time, and sometimes it gets carried south. And so there is some variation in the position of each individual larva. And that's how we were able to kind of tell, okay, are they orienting towards a certain location? So individuals that drifted north of their natal habitat, the reef that we sent, collected them from, were they swimming south? Or individuals that were south, were they swimming north, et cetera. So there was some natural variation in that. A next step for this would be to actively manipulate deployment conditions, like where they're put, kind of like the hypothesis framework, so that we can actively test those hypotheses,
In the parentage analysis though, did you know where the parental sponge was, like I think the Cassidy paper,
right?
North and South? I don't remember.
Yeah, we knew the different populations. So there's another component of that paper that's the position. So the direction of dispersal wasn't depending on the different populations. So whether they dispersed north or whether they dispersed south from their parents was kind of equidistant. There wasn't a strong signal of like, oh, they're going north with the current, or that kind of thing. Yeah, I guess one more question. Yeah.
Yeah, so the orientation in the disc, I guess, listening to some of the conversation, I'm just wondering how natural do you think that is? I mean, is one of the possibilities that, and it's not a criticism, I can't think of a better way of doing it, it's an extremely comprehensive study, but for that orientation question, is it possible that a larvae is disoriented when it's put in the disc and so it doesn't exhibit the behavior it might normally exhibit?
Yeah, so there's been a couple of validation studies with the disc. So the way that this was done in the past was to take a larva and on scuba, release it, and then have one person staring at the larva as they swam behind it in the middle of the ocean, while the other buddy just hung on to them and monitored the depth so that they didn't kill themselves. So I think the disc is an improvement. But they did these tests where they deployed larvae and followed them, but they also did, paired disc deployments, and they got the same orientation bearing results. So that was a good way of validating the study. One thing to note though is, we're aquaculturing these fish on land, right? And so in their container that we're raising them in, they're maybe not getting the same cues that they would be if they're out, moving around in the ocean in different stages of that larval period, right? There were a lot of things that we did to kind of overcome that. So we actually hatched the embryos on the reef. So there's another fun paper that came out this year where I described whenever you shake these, whenever you shake the embryos, they hatch, and they can hatch really early. So we used that to our advantage, and we shook the embryos to induce them to hatch on the reef. So that's how we obtained our larvae. They had the ability to imprint on those sponge cues as they were passing, out of the sponge, and then we brought sponge water back with us and used that to fill the aquariums that they were raised in. And we also used wild plankton in this study to supplement our cultured plankton, to give them kind of a comprehensive option of prey. So those were a few things that we did to kind of give them natural cues, at least at the start of development, to give them something to maybe find their way back to. And so, beyond that, we haven't really come up with creative ways to raise them on the reef or raise them out in the ocean yet. But maybe that's something we'll have to try in the future.